All right, we are live.
Hey everyone, I want to welcome you to this important debate about endogenous retroviruses. My name is Sam. I'm going to be your moderator for tonight. So if you guys have any questions, you can tag me at Redefine Living. That's 2 words. And so, just real quick, I want to let you guys know if you could hit that thumbs up, it'll really help with the algorithm. And then, of course, leave comments after the video. That all helps too. So I guess over on our right, we have Luca. You want to take a moment to introduce yourself?
Yes, as most of you know, I'm an atheist. I have a degree in chemistry. I work as a teacher, and I recently opened a very small channel, and that's all. That's me.
All right, thank you. And then definitely someone who's no stranger to the channel because it's his channel. Go ahead, Donny.
Sam, thanks for doing this, brother. You nailed it. I'm going to be out of a job soon. You're so good at being a host and a moderator. So, yes, my name is Donny B. I'm the founder of Standing for Truth Ministries. In terms of formal debates, I believe this is my 98th. I'm excited to be debating Luca, who's well-studied and well-educated in these areas, being a chemistry teacher. So I do appreciate you doing this, Luca. I've written a book specifically on this topic, the Endogenous Retrovirus Handbook, which Luca has assured me that he's read it all. I've also written numerous other books, and I think as a ministry, we've written close to 20 now. So looking forward to this. Sam, thank you for being a host and moderator. And Luca, thanks for being willing to engage in this debate.
Yeah, my pleasure. Thanks for asking me. All right, so really quick, the format, we're going to have 15-minute openings, followed up by 8-minute rebuttals. And we're going to have a 40-minute discussion, which will be a dumpster fire, I'm sure. We're going to have 5-minute closings after that, and then we're going to have 25-minute question and answers. So again, tag me, 2 words, Redefine Living. Be patient with me. I'm going to try to be saving these comments. So so I guess with that said, unless there's anything else, who's going to go first? So was that, Luke, are you going to go first?
For me, it's the same. Donny?
Yeah, well, I mean, being the title, Are Endogenous Retroviruses Good Evidence for Evolution? Luca says yes, I say no. So, Luca, we'll have you start.
All right, so I think what I'll do, unless you don't want me to, Luca, I can let you know at 14 minutes. Then you'll have 1 minute to kind of finish up, or just go right to 15 minutes, and then we'll be gracious.
I do have my clock here, so
okay.
But I do appreciated.
Cool.
So,
all right.
Well, if we are ready, I can share my screen and start my presentation.
And let me remind people, Redefine Living, you might have already said, for the Q&A portion, make sure you're tagging at Redefine Living, not at Standing for Truth. And then that way he won't miss it. So, Luca, when you want to share screen, let me know. I'll get it up there for you for your 15 minutes.
Okay. Okay. That's the end of the the presentation. You all can see it.
Luca, you're giving us a sneak peek into the after credit scene before the debate even starts.
Yes.
Marvel style. Okay, whenever you're ready.
Okay, so debate, are endogenous retrovirus evidence for evolution? Of course, my answer is yes. So, what's the evidence? Well, we have those sequences in our genome. They look like remnants of viral infection, and they follow a pattern, cladistic. On these things, I think that me and Donny would agree without a problem. So, where we do not agree. Evolution, we are results of Asian infection, and they reflect evolutionary pattern, the cladistic. For creation, ARVs are designed by a creator, and they reflect a design. So those views are equal. For me, they are not, but we will talk about it a little bit more. So, the Weige hypothesis. I do not know the pronunciation of these things, but we will talk about it. So, using the words of the author of the paper, we will see those are Weiges. So they are elements in our genome. They, for this author, they precede ARVs, and they are the explanation for their existence. So not a viral infection, but a unit of DNA designed by the creator. A unit that is the origin of viruses in the views of this scientist. So here you can see a small part of the paper, and I want to point out the style of this paper. This is one of the pictures, and it does represent what will be the evolutionary view of the process. And I think that it's not really something that a real scientist would use. I think, first of all, the terms used are for sure made by are for a public of younger creationists, not for scientists. It's not for scientists, this paper, and I think that it's wrong on a couple of things. You cannot have a substitution like that. I do not think that any scientist will propose anything like that. But it's not my real problem with the paper. Here you can see the proposed mechanism for evolution, and here you can see the mechanism proposed in the paper for the existence of ERVs for the creation worldview. Let's say that. As you can see, those units in this hypothesis are already in the genome. Here you can see they are already designed like that, and that's the hypothesis from this paper. Why I have a problem with it? I will say that this hypothesis could be right, but I have a specific reason to do some criticism on it. So here you can see my points. That would be a good hypothesis if we can show the intact VIG, maybe not all of them, but at least some of them. And how will it be possible to show something like that? Well, if the young Earth creation narrative is true, we could find DNA of ancient humans, or at least humans close to the flood, so less degraded humans, and we would be able to show these elements in our genome. So yes, also to show the actual presence of a designer would be optimal. Let's say that. Let's see on the secular side what we have. So, of course, for what I can tell, ARVs are traces of ancient infections. So why we think so? It's possible to show a mechanism that leads to the formation of ERBs. So we can observe this kind of mechanism. And of course, if we can show instances, example of this happening, and to show these elements to be integrated in the genome and to be kept, to be inherited by the descendant of that organism. So yes, we can, but it's not like I will just tell you so. I did bring some example. So first example, it's from citation, a quote from Dennis' book, more like one of the paper cited in the book. And yes, scientists did observe de novo insertion of ARVs, and they also observed the degradation after the inclusion in the DNA. It's a very interesting thing if you think about that. Also, integration of transposon, so not ERVs, but transposons. Transposons can be the result of the infection of something different from virus, like bacteria, but they can also be from viruses. And that's an example from my previous debates. Basically, the insertion that led to the formation of corn, very peculiar example, but I think it's a good one. Why not using it again? I think it's a good one. Last one. I do not bring any citation for that, and there is a reason. I do have some experience with those kind of things, so I can talk about GMOs in some details without a citation. And it's important to note that we do understand this process, the integration of transposon or ERVs so well that we can use this process to do GMOs. So it's a process so understood to this point that we can use it to our advantage. So that's a very good point from Donny. I want to admit it. So I was pointing on my previous debate with him that LRVs, long terminal repeats, were basically non-coding pieces of DNA. But it's true, they do have a function time to time. They can promote the expression of some genes, and some are really, really important for us. I think that Donny gave plenty of examples about this. So these look very good for creationists. Why I say the opposite? Why? Well, let's look at some citation, some quotes, sorry. This first quote is from the book, the Endogenous Retroviruses Handbook. And you can find here the page where I did find this citation, this quote. So it's a young Earth creation quote. It it's not from a secular scientist. And the view of the author, of course, is similar to Donny, but I want to point out that even them say that most of ERVs are defective retroviral copies. And now we get to a paper. This paper is cited, is quoted in the book, so we can say it's still a quote from the work of Donny. And yes, Donny is right. We do have elements that can promote gene expression, and they are very important to us. But most of those cites, those function are in intragenetic places, so they do not work as intended. They are not aiding us in any way, and they can be also dangerous time to time, actually. So it's very important to note this. Not every ERV is useful. Most of them are defective, or they are in in places where they do nothing, basically. Let's see. This is my last argument. Cladistic. You all know for sure what cladistic is. Here you can see baraminology. That's a phylogenetic tree from answers in Genesis. I do not know if this source is okay. Maybe Donny has a better one, but you can see there baramin and how lineage work in the young Earth model. And when we talk about my model, well, here you can see the tree of life for my side. Very, very good. It's different. So when we look at ERVs, what we find, and here you can see the result. It's taken from a paper about old world monkeys. We actually are in this clade, at least for my position. So it's quite important. I did not read the paper. I was just interested in the picture. So, conclusion. Here are my conclusion. The Weige hypothesis could be good if you can show evidence. The vast majority of ERVs seem to be inactive or damaged. Cladistic evidence, for what I can tell, does support my side. So, at this point, I do not see any reason to think that Donny's position is reasonable, but I'm open to being wrong. Thanks for watching. For any question, contact me. This is my YouTube channel, very little, but it's growing. And I hope to see you soon.
All right. Thanks, Luca. Good job. You even got a comment on your lab coat. They said you should have a beaker behind you, maybe. So, yeah, so I just want to remind everyone to tag me, redefine living, 2 words. I will be looking in the chat, though, so if you do ask a question at Standing for Truth, I'm going to try to grab those too, but no promises. So that said, Brother Donny, 15 minutes, whenever you're ready.
All right. Thank you, Sam. Let me just get my slides up here real quick. And Okay, here we go. Can somebody share my, just kidding, I'll share my own screen. So here we go.
Hold on, let me do that real quick.
Yeah. Love it. Okay. I'll hide this. Let me know if my audio and everything's coming in nicely.
Yeah, just go full screen maybe, but everything sounds good.
All right. Let's have some fun. I'll start my timer here. Luca, thanks so much for your opening. Lots of interesting points to address. And as a matter of fact, several that I'll address here in my opening statement. So, endogenous retroviruses, evidence for evolution. Donny B versus Luca Medugno, debate number 98. I've written a book on this, as I've pointed out earlier, and I answer all of the common questions, challenges, objections regarding this topic. I'm currently working on a follow-up that basically deals with very thoroughly all of the objections and challenges and just the repeating of talking points that I address directly in the handbook itself. I'll be putting that all into a follow-up book, Refuting the Critics. Again, the Endogenous Retrovirus Handbook is available on Amazon, and you can find endless content on our Standing for Truth ministry. So the title of tonight's debate: are endogenous retroviruses good evidence or reasonable evidence for evolution? And firstly, I want to point out that endogenous retroviruses, what are they? Well, they're very small pieces of DNA that are found within the genomes of many types of creatures. And the most important question to ask is, are these DNA elements the ancient remnants of past viral infections? Or are they created units of DNA function? And this is the question that my opening presentation will answer. Now, firstly, when it comes to the word evolution, okay, are endogenous retroviruses good evidence for evolution? Well, what do we mean by evolution? If by evolution you mean dogs and wolves are related, well, we're not going to have a problem, okay? Because evolution, biological evolution, just simply means changes in allele frequencies in populations over generations, okay? Change over time. People say, you know, phones have changed over time, computers have changed over time. I'm not going to have any issues with that. But if by evolution you mean that dogs, chimpanzees, humans, banana plants, and strawberries are related, well, that's the kind of evolution that we're going to have a disagreement over. That's the kind of evolution that is not scientific, and that is the one we're essentially debating here tonight in context of endogenous retroviruses. So the separate ancestry model, as compared to the universal common ancestry model that would say whales and strawberries are related through common ancestry, would suggest that God created distinct kinds of animals. Okay, the Bible clearly tells us that God created Adam and Eve separate from any other form of life. So here's a basic diagram that you can find in a must-read book, Contested Bones, where we have humans separate from all other forms of life. Okay, I can stay as close to home as possible and demonstrate through various genetic related arguments, like the Y chromosome dissimilarity between humans and chimpanzees, mitochondrial DNA mutation rates, Y chromosomal DNA mutation rates, DNA function, linkage blocks. I can demonstrate that humans are not related to any other form of life, specifically not related to chimpanzees. So if humans aren't related to chimpanzees, then that means they're definitely not related to strawberries and whales. So our model would suggest that in the beginning, God would have front-loaded the original created kinds, okay, humans, and then this would obviously apply universally among the original created archetypes. He would have front-loaded them with what's called created heterozygosity, which is just a technical way of saying design diversity. Okay, heterozygosity is a state of DNA diversity, and this makes sense both theologically and scientifically, because when God said to be fruitful and multiply, he didn't mean that to mean, you know, be fruitful and clone yourselves. So pre-existing heterozygosity makes sense, but how this applies to the endogenous retrovirus debate is the fact that God would have also front-loaded various classes of DNA elements, functional DNA elements, ERV sequences being one of those, which we're going to touch on here, okay? Also, since the DNA differences and DNA diversity is built in, okay, we don't need millions of years to explain the origin of species and new chromosomal combinations, because simply through processes like recombination and gene conversion, you can get a great deal of morphological variability because there's an amazing amount of morphological adaptability built into each created kind. So just through the shuffling of these pre-existing DNA variants, you can get new combinations of chromosomes, allelic variability quickly. Okay, so endogenous retroviruses, let's go over some of the details. Okay, firstly, I want to point out that when it comes to the structure of ERVs that Luca touched on a little bit, okay, the structure of ERV sequences, they match modern retroviruses. For example, think HIV, okay, where you have, I want to go to a diagram that I put in here, where you have the LTRs on both ends of the retroviral DNA. You're going to have identical sequences known as the LTRs. Okay, those stand for long terminal repeats, which we understand are functional. In between the LTRs, we find the GAG, the pol. Now, the pol is what codes for what's called the reverse transcriptase. And then we also have the ENV, which is the envelope protein that codes for the envelope that makes up the body of the virus. And these structures are common in ERVs and retroviruses, which is precisely why evolutionists believe they are the ancient remnants of past viral infections, since they share so much similarity to exogenous retroviruses. Okay, and this is what I am going to want to touch on. Okay, so because Luca, evolutionists like Luca, assume that these ERVs are the ancient remnants of past viral infections, and they consider them to be genetic fossils that point to common descent, they will say, as a main line of argument, that the chances of two ERVs being inserted at the same exact location in separate organisms, let's say humans and chimpanzees, is very small. Okay, they will argue that the chance of a human and a chimpanzee being infected in the same exact spot by the same specific type of virus is far less than 1 in 10 million. And to them, this is highly unlikely. And of course, the more shared ERV sequences, the more unlikely it becomes that they're inserted independently. But again, that whole argument from statistical probability relies on the assumption that these really are the ancient remnants of past viral infections, as we can see here. Human endogenous retroviruses are expected to be the remnants of ancestral infections. Okay, so they represent 8% of the human genome. They are an important component of the human genome. According to the evolutionary model, they're incorporated into the genome through the invasion of infectious exogenous retroviruses that basically would have happened millions of years ago. They look to them as fossil viruses. And again, for the most part, the evolutionary community will admit they occurred millions of years ago. Exogenous retroviruses are relics of the exogenous retroviral infection of germ cells that result in integration of what's called proviral DNA into the host genome. And I've got a whole bunch of points in terms of the details of endogenous retroviruses. What I want to move on to the important questions, okay? The important questions that I'm going to answer are, why are there ERV sequences shared between the genomes of organisms? If viral-like sequences in the genomes of organisms are functional, why do they bear similarity to viral genetic material? What is the best explanation for the nested? So they'll look to what's called a nested hierarchical distribution of these elements, where more ERV-like sequences are found in similar locations between a human and a chimpanzee, and with fewer ERV-like sequences shared between what is assumed to be a more distant relative, like gorillas, the orangutan, or even mice. Okay, so, Luca would look or appeal to what is called a family tree, that he would interpret the nested hierarchical patterns, let's say within primate genomes, in terms of their arrangement of these ERV-like sequences as reflecting common descent rather than separate ancestry. So I want to answer that question as well. And of course, can the separate ancestry model explain the data better than the common descent model? Okay, so here's an important detail. I want to focus on the placenta for a minute, okay? In the early stages of development, humans will develop what is called a placenta. And a placenta is a temporary organ that arises during pregnancy. This structure is absolutely vital. It's critical for ordinary and healthy pregnancy. It provides nutrition to the developing baby. It also helps it get rid of waste. Now, for the placenta to work, though, this important structure needs to be connected to the baby. And an endogenous retrovirus is actually key to embryological development and placental function. The placenta secretes a protein that binds it to the embryo, which keeps the two connected for the next several months of development. We literally couldn't exist if we did not have endogenous retroviruses. I like to exist. Our award-winning moderator, Sam, here, I believe, likes to exist. Luca likes to exist. Okay, so if we have shared sequences that are involved in embryological development, well, guess what? Chimpanzees also require embryological development. They require DNA elements functioning and determining cell types, gene expression, gene regulation, okay, acting in cell stress responses. So, of course, we're going to expect shared sequences if these really are created units of DNA function, okay? So the DNA that makes up that protein is very similar to a region of a retrovirus that allows the virus to attach to its host cell. Without endogenous retroviruses, the placenta, which is critical to pregnancy and embryological development, would not work. We would basically not exist, okay? Because these functional DNA elements are essential for life. And endogenous retroviruses don't just assist in embryological development and embryonic immune systems, they also work in our immune systems after we are born. Our immune system pathways are dependent. They are literally dependent upon enzymes generated by endogenous retroviruses and ERV-like elements. These so-called ancient remnants of past viral infections are safeguarding our bodies against microbes and other viruses. They're also participating in allowing for small-scale but significant changes in how the human body works in general. As a matter of fact, these many functional roles, okay, many of the functional roles of ERV elements and retrotransposons are basically dependent upon their sequence similarity to exogenous retroviruses, the LTRs, the GAG, the POL, the ENV elements of it. Those are necessary components of the ERV sequence in order for them to be able to carry out their job. Okay, this also comes down to the what's called the junk DNA paradigm, because ERV sequences have been, and still for the most part, are assumed to be junk, evolutionary leftovers, genomic fossils, viral fossils, okay? So with the overturning of junk DNA comes the overturning of endogenous retroviruses as evidence for common descent. And we understand, actually, that most of our genome has tremendous evidence for biochemical function or activity. And it was in 2012 that the ENCODE project came out with remarkable findings indicating that upwards of 80% of the human genome was active to some extent. Okay, and of course, the evolutionists have jumped all over that and criticize it. They need to maintain junk DNA. Okay. And I address all that in my book. So here's just a few of the functions of non-coding DNA: regulating expression of genes, regulating genome expression, maintaining genome integrity, work as regulatory elements during transcription and translation, controls the process of mRNA and protein formation, repairing DNA, aiding in folding and maintenance of chromosomes, controlling RNA editing and splicing, not predicted by the evolutionary model. Okay, acting as a mutational buffer, maintaining 3D structure. Pseudogenes, we now understand, are necessary to sustain healthy life processes in the cell. ERVs, again, ERVs are involved in antiviral function, tumor suppression, gene regulation, which I do want to touch on. Okay, so again, it turns out that one of the major functional responsibilities of ERV sequences is that they operate in the innate immune system. They literally perform an antiviral role. And I want to go to a paper here specifically right here. Switching sides. How endogenous retroviruses protect us from viral infections. Okay, so they literally are our built-in antiviral programs. They perform an antiviral role, and they are valuable DNA elements that work considerably in the immune system of their host. The way that they systematically exercise their antiviral effect has to do with their sequence, resemblances to viral material. So again, without the nature of the ERV sequence, the way that they're designed and structured, they could not actually carry out the significant job that they perform. We should be thanking God that they resemble exogenous retroviruses, okay? The properties of an ERV sequence are absolutely necessary considering their fundamental roles in aiding the immune system. When it comes to explaining the functional roles associated with herb elements, okay, all we get is rescue devices, storytelling, and imagination. We had a PhD virologist, Dr. Dan Stern Cardinal, admit that they've never actually observed in real time, empirically, there's no technical paper showing a non-functional endogenous retroviral-like sequence going from non-functional to something incredibly functional in determining cell types, in regulating genes, in the embryo, placental development, so on and so forth. So what I say to these rescue devices is cool story, bro. So as I come up to the 15-minute mark, time really flies by. Last thing I want to point out when it comes to nested hierarchies is, as we can see, the evidence best suggests these are created units of DNA function. So if we share more with the chimpanzee than we do with a mouse, there's no surprise there, since humans and chimpanzees share more genetically, in physiology, in morphology and anatomy anyways than, let's say, between a human and a mouse. So and that's 15 minutes. Time flies by, and I am going to wrap it up there. Thank you so much.
All right, thank you. Yeah, I kicked myself out, so you might have to take away that that award. So yeah, just want to remind everyone to tag me at Redefine Living. I am looking very carefully over the chat, so if you guys do tag Standing for Truth, I'll still grab that and save those questions too. Try to make sure they're relevant if it's possible. We've got questions for you, Luca, coming in, asking if you're married, what do you use in your hair, and things like that. So, so with that said, Luca, you ready for your rebuttal? You gonna rebut all that? You got 8 minutes whenever you're ready.
Okay, give me a second. I set my lock. And okay. So it was a good presentation, and I do have a lot of questions, and I do have an argument to make. So Donny is right. A lot of ERVs are really, really useful, and they do resemble viral infections. So that's a chicken or egg problem. It can first be or an infection from a virus. And here you need to bring the evidence, because in my presentation I did bring up some evidence for what for my story. Let's say that I'm personally, I did work with ERVs, not really ERVs. I did work with transposons because I did create GMOs when I was 18 years old, more or less. So I am familiar to the process. Of course, I was in my high school years. I was not a doctor. I'm not a doctor even now, so I was very young. But I did those kind of things. And it's very important to see what's the evidence for an hypothesis. We do understand how transposons, so ERVs, works. We do understand so well this process that we can use it to our advantage. I don't know if you know, but we can create animals with human organs and things like that, plants with in part bacteria that can kill infestant just like that. They produce their own poison to kill off insect and things like that. So we do understand the process. And we do have evidence for ERV's insertion. I cited corn. but we have more example for it. For example, I did talk about the sweet potato plant long time ago, I know. Also, the formation of beneficial mutation on ERBs. I think that the lizard example that I used with Kant was a good one. We are observing the formation of a placenta, and it's quite relevant because Donny was right. The placenta is fundamental for us, but what I want you to understand that not every creature need one, and we are observing creature evolving a placenta, and they have a placenta, they have not a placenta, they have something in between in the same species. So it's a process, and we are observing it in real time. So I want to focus in my discussion over these things because it's important when you have a hypothesis. It's important to bring evidence, and if I put myself in the mind of a creationist, I will be thrilled because I have an hypothesis. ERVs come from this design sequence. Good. I need to find evidence for it. And if young Earth creationist view is right, we have the means to show that's true. Because if the flood was just 4,400 years ago, we can find the DNA of these people. It would be very, very devastating to my position to find Noah's body or things like that. Just think about it. So I will wait for something like that. It will be very, very exciting. And if you are a creationist, I would advise you to put yourself on the line and go and find evidence. Go to the Middle East, maybe not now if the time it's not good. We are almost over with a pandemic, but it's not a good time to make travels. But I will be very excited if I will be a young Earth creationist. We have an hypothesis, let's find evidence. I do not say that. For what I can tell, the evidence is still on my side. On cladistic, it's very important. Donny is right. We would expect some kind of clades on both models, but a designer would not need to follow evolutionary patterns. And that's my problem. If you look at ERVs, they give you a very secular view. They follow what evolution will be. And I think it's a problem. I can stop my rebuttal here.
All right. Thanks, Luca. So, so Donny, are you ready for a rebuttal?
Oh, I'm ready. I'm
All
ready.
right.
This is
Well,
gonna be
you
fun.
got 8 minutes sharp. All right, go ahead.
All right, I'm going to start my timer now, and I got a lot of visuals for this, and this is going to be good. So here we go. Luca started off. He talked a lot about the VIGE, which stands for variation-inducing genetic element hypothesis, which is supported by the scientific data. I've got paper after paper demonstrating that these transposable elements can actually generate variation, novel variation, through their ability to retrotransposition. to transpose throughout the genome, okay? So he finds it unconvincing, but the hypothesis, the model is supported by the empirical scientific data. Problem is, Luca will have to show us how his position can explain the origin of retroviruses to begin with, since retroviruses require a host to replicate. It makes sense that they originated from within the genome of their host, of their respective host, since again, retroviruses require a host to replicate. Evolutionary scientists actually recognize this, which is why a lot of them, and I'll show slides on this, believe in what's called the escape hypothesis, that retroviruses have escaped from the cell rather than originating from outside, okay? Luca talked about the koala retrovirus paper, which I'll get into as well. That is the one, degradation and remobilization of endogenous retroviruses by recombination during the earliest stages of germ line invasion. I wrote a lot about that in my book. Talked about transposable elements in corn, and it's no evidence for Luca because, again, these transposable elements can be turned on, right? They're suppressed due to DNA methylation. They're turned back on, they move around, and they can lead to variation. They can lead to novel phenotypes. That information's built in, though, okay? A lot of these DNA elements, they're in latent form. They're waiting to be revealed and manifested outwardly. Luca talked about function in intragenic regions, implying that they don't aid in our health in any important way. Well, I'm going to need him to expand on that one because, as I went over in my opening statement, ERV sequences, without them, we couldn't exist. That's pretty essential. Okay, they're involved in embryological development. They're involved in, obviously, the placenta, determining cell types, gene expression, antiviral protectors, cell stress responses, so on and so forth, okay? Some are harmful, yes, since mutations can take a good ERV sequence into something disease-causing. Some jumping genes can jump into the wrong place, and that will result in disease. For example, if one of these transposable elements were to jump inside a functional gene, that could lead to harm, okay? So I do want to point out the argument from the koala, for example, because as we know, retroviral sequences, they have RNA as their genetic material. And what they can do is they can reverse transcribe this into DNA, okay, which is necessary for integration. And that's how they get incorporated into the host genome. Some endogenous retroviruses can make copies of themselves and insert throughout the genome. All right, they can literally move around. And this capability of moving around the genome actually turns out, we read about this in specifically the paper that he mentioned in his opening statement, okay? This turns out that it can disrupt the incorporation of retroviral DNA into the genome. All right, this is a significant function because it can literally disrupt the endogenization process of retroviruses. This is also one of the numerous explanations for, again, why ERV elements look the way they do. Their similarity to retroviral genetic material makes sense because of their functional mechanism. When it comes to the hierarchies, okay, yes, overall, we're going to expect more of these ERV sequences or the transposable elements between, if they're created units of DNA function, between humans and chimpanzees and between humans and a mouse. Just stand back, okay? You can see a relative hierarchy. As a matter of fact, you can see a relative hierarchy when it comes to design modes of transportation. Sedans share more with SUVs than sedans do with, let's say, a tractor trailer or a bicycle, okay? These groups within groups within groups patterns are simply characteristic of design, but you find inconsistency after inconsistency with the hierarchy. Okay, notice this paper, a HERV-K provirus in chimpanzees, bonobos, and gorillas, but not in humans. These observations provide very strong evidence that for some fraction of the genome, chimpanzees, bonobos, and gorillas are more closely related to each other than they are to humans. So even when it comes to the hierarchies, there's inconsistencies. You could look to convergent evolution, again, the Y chromosome, incomplete lineage sorting, so on and so forth. So here we go. Jumping genes generating diversity. The ability of transposons to increase genetic diversity, together with the ability of the genome to inhibit most TE activity, results in a balance that makes transposable elements an important part of the genome. Here's an important part right here. I want to focus on this part of the citation. if the accidental infection, and remember, there's no observable evidence for how this could take place, okay? They'll oftentimes try and say that the ERV elements have contributed new genes, like the syncytin, which they say is a co-opted ENV protein. I mean, how sweet are the retrovirus, right? Hey, host, you know, you can borrow these genes, and with them, apparently now mammals can come about. Because notice, if the accidental infection of a mammalian ancestor by an exogenous retrovirus had never occurred, the placenta and the mammals that produce it, including humans, would have never existed. So Luca keeps saying there's no evidence. Here's evidence. What more do you want? Okay, human endogenous retroviruses, HERVs, have recently been suggested as mediators of normal biological processes. There's a family of retrovirus known as HERV-H, and they strongly influence our growth, development, and our genetic code. Okay, and they have a massive, these ERV sequences have massive impact on embryological development, gene expression, and co-option just isn't going to cut it. Again, all I got to say to this rescue device of co-option or the ERV elements, you know, lent us some of their functional sequences to be highly beneficial in us, growth, development, and so on and so forth. You know, a cool story, bro, because I'm into empirical evidence. Retroviral promoters in the human genome. Scientists identify new beneficial function of endogenous retroviruses in immune response. Literally paper after paper. That's why I wrote a whole book on it. 200 papers or 200 pages with just dozens and dozens and dozens of papers all throughout. Okay. Notice this, although TEs have in general been regarded as non-functional junk, so they'll admit that they've regarded these TEs as non-functional junk. Notice this, studies have revealed that TEs, transposable elements, have had a substantial and sometimes beneficial impact on host genomes in several ways. Overall, our results provide direct evidence for what? Retrotransposons in actively shaping cell type and species-specific chromatin architecture. And again, paper after paper. Notice this one. Far from being junk DNA, the pervasive retrotransposons that populate the genome have a powerful capacity to influence genes and chromatin. LTRs have functional roles. The shared mutations, we understand there's mutational hotspots. If the genome is mostly functional, which we know the evidence suggests, then that means there's more hotspots in the genome that exist. Okay, so there's going to be a lot of commonly affected areas between humans and chimpanzees, since most of the genome reflects functionality. And so, yes, within the ERV sequence, we would expect that even some of the mutations are shared. But that brings me to my final point here as I come up to 8 minutes. Okay. The bigger question is, what is the origin of genetic variation, genetic diversity? Evolutionists look to mutations. We look to created nuclear heterozygosity, which means that which the evolutionist points to as a shared quote unquote mutation may not actually be a mutation. It may just reflect the functional requirements of the respective organisms. ERV sequences are not good evidence for common descent, and that's 8 minutes. I yield.
Good.
All right, great job. So we are going to take a 2-minute break really quick. If you guys want to go get some water, do whatever you want to do.
Yes,
So
it will.
while you guys are doing that, I guess I'm going to sing to you. No, I'm not going to sing to you. So so it looks like John, logical, plausible, probable, is having an after show. You guys aren't going to want to miss that. So if you guys don't mind, go ahead and hit that thumbs up if you haven't already, and subscribe. If you are wondering how you can support the ministry, in the video description, there are a few options. But really the best thing you can do is just by hitting that thumbs up button and, share this video out, these uploads on social media. It really helps with the algorithms, or after these uploads, come back and leave a comment. So, yeah, so there's a few streams coming up. There's a debate between T-Rock, who's awesome, and T-Jump. So that'll definitely be something to watch. We have a debate coming up about 2 Peter. What's the best exegesis of 2 Peter, chapter 2:20? There's a Bible translation debate, a King James verses debate. That's going to be coming up pretty soon. I think later on this week or next week. We have another debate here with Kent Hovind coming up. What is the best evidence for evolution? And another Bible translating debate coming up here also. And yeah, hopefully Professor McQueen won't be too long before he comes and does another stream. You guys, there's playlists. There's tons of content, a lot of stuff in those playlists, past debates. So yeah, go check it out and share it out if you want to help the channel grow. That's what we can do. So with that said, it looks like we are into our 40-minute open discussion. You guys ready? You want me to pick a topic, or are you just going to go for it? I think Luca brought up corn, and Donny, I think you said that was a corny line of evidence. That was more than a corny joke.
Yes, I think that corn is a good one, actually. There is a reason because we think that we can demonstrate that those insertions really happened. First of all, we have archaeology supporting that, because we do know what was the more ancient plant, and that was the Teosinte. And it's important because when we are talking about archaeology...
Actually, Luca, let me just ask you a clarifying question. Do you believe that my model on endogenous retroviruses suggests that there are no true events where exogenous retroviruses make their way into the germ cell and are passed on?
Well, it's possible to have both, but if we are talking about my model, we have a mechanism to insert those. If we are looking to your...
well, in my book, Luca, just for another clarification, I have a whole section on fixed herbs versus unfixed herbs. So herbs that are pre-existing and created versus herbs that are the result of the infection of an exogenous retrovirus that infected in the reproductive cell lines and was passed on. Do you remember what that prediction is?
Yes, that we will not observe fixed ones. And that's my point. The one with the Teosinte was fixed. And we can demonstrate that was a real event, because if you look to archaeology, we have a sequence, and we do know where
are you saying the transposable element in the maize example was fixed and functional?
It got fixed after the event.
Okay, so it got fixed. Is it functional?
It wasn't. Yes.
And you watched it in real time get fixed in corn, right?
No, we did observe the process through archaeology, because if we know where we start and we know where we get, we know that event take place.
Okay, but this jumping gene, this transposable element in the maize, in the corn that led to variation, it had a pre-existing function?
It was not there.
Because I read through the paper and it looked like the transposable element was silenced. Because remember, a lot of these transposons, through DNA methylation, they're silenced or turned off. But due to environmental trigger or stressor, the appropriate stressor, they can be activated. They can, a lot of these retrotransposons have a promoter in them, and so they can move around the genome from one chromosomal location to another, generating function, generating variation that can be passed on.
Yes, but you need an infection to have that element, and we know that take place. If you don't like maize, we can talk about this with the plant. In that case, we also know the kind of bacteria that infected the plant. We know so well this process that we design GMOs from it.
And for the audience sake, GMO stands for genetically modified organism. So, right, we have human intervention here, human manipulation. But I'm curious, how does this help the case for ape to man evolution, for humans and chimpanzees sharing a common ancestor, right? How does this help the case, since plants, as far as I know, don't need functional DNA elements for embryological development? Plants oftentimes have a lot of variation, built-in redundancy due to the environments that they exist in. There's a lot of silenced transposable elements that can be turned on for adaptive episodes. But when it comes to humans and chimpanzees supposedly sharing a common ancestor, okay, how does that help your case there? I mean, where do we see in real time? Go ahead.
I'm using the examples that I'm more familiar with because I worked with GMOs and I was in a school that was teaching agriculture. So those are the example I'm more familiar with, but we do have example of those even in mammals and things like that. Of course, plants are a good one because we can trace the infection. In the case of the sweet potato plant, we also know the kind of bacteria, so we do know the process. And they are still eukaryotes, so they share a lot of process with us. So it's not that far-fetched example.
Okay, so let me.
Of course, we are same. Yes.
I just don't find the example from maize. I read the paper. It's not convincing to me because, again, DNA methylation. Okay, the epigenome can essentially keep a lot of these pre-existing functional transposable elements silenced until they need to be called upon through environmental stimuli and environmental stressors. But when it actually comes to, let's say, mammals like us or the chimpanzees, because typically the evolutionists want to say that chimpanzees and humans are undoubtedly related because of shared ERV sequences or shared ALU sequences or shared, these various classes of retrotransposons. So I'm very curious what your best example is over the last few months of you reading my book and studying. Okay. An example like this, where these HERVs, human endogenous retrovirus, are playing important roles in what? Embryological development, where notice this. If this supposed accidental infection of a mammalian ancestor by an exogenous retrovirus had not occurred, the placenta and the mammals that produce it, including humans, would have never existed. I want to see a real-time example. Okay, take the lab. I mean, you have mice, subject mice retrotransposons to mutations or whatever it takes, and show me a real-time example of one of these endogenous retroviral-like sequences going from non-functional or just altering their pre-existing function to the point where now without them, we couldn't exist. Can you provide that and take your time, Luca? I know I said
yes, the lizard one. We are observing a lizard developing a placenta, so it's a very relevant example.
Wait. I'm talking about endogenous retroviruses or other classes of retrotransposons.
Yes, we are talking about that because the sequence is very similar to that, the syntychin one. So yes, we are doing this kind of discussion and we are observing that kind of change in real time because those lizards, as I said to Kent, they are.
Well, they're not evolving placentas due to endogenous retroviruses, though.
Yes. And so we can observe how they got integrated. We do know the process so well, we can use it.
Is it your position that an endogenous retrovirus integrated into the lizard genome and became functional in the embryo and in placental development?
I cannot remember the exact event, but I remember that was
right. Because I've read through all those papers, and what it looks like is within. And here's I don't want to spend too much time on it because again, it's not endogenous retroviruses that are involved in these adaptive episodes. But it looks like, I believe it's these variety of skink species. And within the skink species, depending on their environment, they actually have a dual reproductive system where they can hatch eggs, or they have function in terms of a placenta. And depending on what environment they're in, that will determine what reproductive system is manifested. It's kind of like how GM builds their cars with both a heater and an air conditioner. So if I'm driving a GM vehicle in Alaska, well, I'm probably going to have the heater on predominantly, right? Dominantly. But if I go to the deserts of Australia, then I'm probably going to use the air conditioner, because cars are built with dual temperature control systems. So once I turn on the air conditioner in another environment, the evolutionists are going to say, look, this vehicle is evolving the ability to have an air conditioner. No, it was already there. It was just turned off, and now it's being turned on in the appropriate environment. And that's what we see with these so-called skink examples of a placenta. And you can respond to that. But again, it's not anything to do with endogenous retroviruses. So I do want to make sure that you're...
This is the case why we do have example in the middle with a partial placenta, because we also have that.
Well, because if a certain skink species is predominantly using one reproductive mode, well, what happens over time is the other reproductive mode is suppressed to the point where they can even lose it. So you're saying this is something in the middle, when in fact it's an example of loss. So you know how they say if you don't use it, you lose it. So just like if you have a vehicle that you're just mostly in Alaska or some really cold conditions, you're using the heater all the time. You're not using the air conditioner. And over time, that air conditioner, due to underuse, might actually break down. It's already there, but either one can be revealed more, or to the point where one can even be lost. So, but you know, and we can agree to disagree there, but I really, Luca, just want to see an example of an ERV element that is, I guess, a basic question, and take as long as you need. I know I'm trying to get as many clarifying questions as I can. Okay. An ERV sequence today, you can go in the lab or observe it in real time. Doesn't matter. You know, you even use, mice. I want to see an ERV element, okay, an exogenous retrovirus that is integrated in the germ cell line. It's been passed on. What the evolutionists typically say, as you know, Luca, is over millions of years after these infections occur and they're integrated, these sequences will be cleaned up through mutations, and then the infectious part of the retrovirus will be eliminated, right? And oftentimes they'll say, like in the case of syncytin, that the ERV elements will contribute new genes that can be utilized by the host for functional purposes. I get it. It's a nice story. I talk a lot about it in my book, but show me a real-time example. Show me empirical evidence of this actually happening, of an ERV sequence going from non-functional or altering its function or donating its pre-existing properties to function in the embryo, to function in determining cell types, to function in the immune system. So that's all I'm asking for, Luca, and take as much time as you need to answer that. Go ahead.
Okay. So if you want a nice example of an experiment, we can have any GMOs, because the process is that process. Basically, you get an element. Of course, you modified it to pick up the sequence, DNA sequence you need, but it's still this process, and you put it in the organisms. So the best one to do that are obviously plants, but you can do it even with animals. I've seen.
Are you saying it's been done in animals? They've genetically modified an animal to the point where a pre-existing ERV sequence became functional?
Well, yeah. No. They put an entire sequence inside the pig, and the pig changed, and the change were inherited. So we know that process can work.
Well, in pigs, I just finished a lecture series in virology where the lecturer from, I think, Columbia University, he pointed out that they're removing the existing ERV sequences. I don't know if this is what you're talking about. I'm just trying to follow all of your arguments, Luca, so I address them all. They removed all the pre-existing ERV sequences to see if they're there for a reason. And it actually turns out that yes, there is offspring, pig offspring, but they are in an unhealthier state than they were prior to the ERV sequences being removed. Is that the example you're looking to?
We are talking about something different, because what I've seen pigs so modified so much that they start growing human organs inside them. So I've seen.
Okay, so is it your position that through genetic modification, that there's a real-time example?
Yes, and how you do that? You insert ERV sequence into the pig, and the pig will.
Okay, I just want to follow you. So humans insert an ERV sequence into a pig, into the pig genome, and then what happens?
Transposons, more likely.
Okay, so a transposable element, they integrate it into the pig genome, and then what happens?
Yes, they start growing human organs inside them, and I've seen evidence for it with my eyes. So we know that process can work because we do all the time.
I don't see Luca, I'm sorry, because we have limited time. I got to get slightly aggressive. I don't see how this example of human intervention through genetic modification, apparently they're inserting, and I'd like you to send me the paper so I can read it myself, where they're inserting an endogenous retrovirus into the pig genome, and now subsequent generations, we have pigs growing new organs, is what you're saying?
No, different organs, because what they were designed for, they were designed to supply fresh organs to donators. So you will grow pigs with organs.
Well, did you want to share screen and pull this up so I can see it?
I cannot pull this up because I've seen it with my eyes, and it was quite a long ago. I do not have a paper right now.
I'd like to see
that I've seen those things myself.
Okay, so pigs already have the ability through their pre-existing retrotransposons, right, to produce a baby pig from zygote to baby, okay, based on their functional DNA elements. So if a scientist is now manipulating the pig genome for some kind of desired outcome by inserting an ERV sequence into it, which again, I want to see this paper. Okay, well, I don't understand how this genetic modifying example helps your case. I mean, we see that the evolutionists are saying millions of years ago, okay, the accidental infection of an exogenous retrovirus that was infected in the right way to be passed on to subsequent generations. Okay, eventually mutations cleared out that the infectious part of it, and then somehow adopted a novel function in the embryo to the point where we couldn't exist. This pig example, well, the pigs are already existing. So whatever happened through genetic modification is not creating anything new where now the pig needs to survive. And this is the example that I'm asking for. And I've already heard from PhD scientists that these examples don't exist. We have to infer it, right? Like saying, well, the endogenous retrovirus contributed new genes like syncytin for the embryological development. So, you know, that's all I'm asking for is a real-time example of an endogenous retrovirus viral-like sequence becoming functional to this extent, essential. Go ahead, Luca.
Okay, as I said, you cannot use a single example. It will be very unlikely to observe something like that in nature. What we can do, we can observe things happen in the past. I think that the sweet potato plant is one of the best example for it, because we do know what happened. We do know what was the infector. Sorry, maybe it's a wrong term. Uh, it was an
But you just admitted that you never seen this transposable element integrate. You're inferring it from, correct me if I'm wrong, you said,
Let me finish, Donny. What I'm telling you, we can observe this process in the past, and we do know enough of this process to replicate it today and doing things like GMOs. That's how well we can understand it.
yeah, I'm just going to have to, we'll have to leave it up to the audience. I don't find an example of GMOs, genetically modified organisms. And again, we need to see the paper. I want to, look into it a little bit. But even with what you're saying is true, that's not an answer to the question that I'm asking. It's not impressive that scientists, I mean, we see it with the Phoenix virus experiment, where through manipulation from scientists, they were able to produce a virus from a deactivated virus. Okay, well, I mean, yeah, humans through human intervention, we can do a whole lot of things. That's not showing that it happened in nature. It's also not showing how a non-functional endogenous retrovirus, through a series of mutations, can become something as important in determining cell types or embryological development. My question to you here, I'm not sure if you're familiar with this paper, inconsistencies with the hierarchy, a HERV-K provirus in chimpanzees, bonobos, and gorillas, but not in humans. So although there is a hierarchy generally, we do find certain DNA elements in their placement, the way that they fall out into, where it looks like, apparently, according to this distribution or this family tree, chimpanzees, bonobos, and gorillas. Okay, so these observations provides very strong evidence that for some fraction of the genome, chimpanzees, bonobos, and gorillas are more closely related to each other than they are to humans. But even though chimpanzees are supposedly our closest cousin, so there are some inconsistencies in the hierarchy. Luca, what are your thoughts on that?
Well, I do not know this example in particular. I do know that time to time we get those kind of things. Science is all about finding new things, so we need to improve step by step. When we find a problem, we need to find a solution or a better hypothesis. I do not know what the proposed explanation for this inconsistency is. But for the most part, you will agree with me. ERVs paint out a picture that resembles a lot my theory.
No,
So,
because again, because as I've pointed out, without retroviruses here on my slide, we would not have placentas and we would not be bearing live young. We would not exist. ERVs are expressed during human embryonic development. We understand that they're functional again in the immune system. If we didn't have endogenous retroviruses, the pre-existing ones, I mean, we see it in that koala example, that we have new endogenous retroviruses, and what the pre-existing endogenous retroviruses are doing is they are, through retrotransposition and recombination, is they are disrupting the new endogenous retroviruses and disabling them, so they're not harmful anymore. So we have, again, these pre-existing fixed ERVs acting in the immune system, acting to prevent exogenous retroviruses from becoming endogenous. Or if they do become endogenous, then those pre-existing functional ERVs are working to disrupt or disable them from causing harm. So therefore, if we find a relative hierarchy in, say, primates, that's no surprise. Because if they're created units of DNA function, of course, humans and chimpanzees are going to share some. But I just showed you a paper where, although there is a relative hierarchy, there are inconsistencies to the hierarchy as well. And that's the problem I have. Go ahead, Luca, take your time.
Okay, so I want to ask you a relevant question on that. I do not know how much we have to end the discussion, because I want to ask you one particular thing. Because
go ahead.
Okay, first of all, so we do observe novel insertion in koala. That's good. And they change the sequence of those ERVs. You are familiar with the structure of proteins?
Yes.
Okay, so it's possible if you scramble the information inside those pieces of DNA to get protein with a different form.
Can you repeat that last part, Luca?
If you are scrambling those sequences as written in the paper, you can get to a pseudogene or something like that with a different shape of a protein. If you are scrambling the content of the string of DNA.
If you're scrambling the string of DNA, you are going to, what's your question?
If you can get a protein, maybe a less bigger one, but with a different shape. I was asking if you were
a variation of
familiar with the structure of the protein. When you build the protein, you have the sequence, then you have the secondary structure, alpha or beta sheet, and then you have bonds with sulfur. I recall.
Right, but Luca, how does this help your? Okay, but what's the relevance to this? Because again, we have limited time, and I really need to. You're in the affirmative tonight. Are endogenous retroviruses good evidence for evolution? And
yes, it's relevant.
I don't see how it is, because I don't see you addressing any of the differentiating lines of evidence here.
Because if you are scrambling those units of DNA, you will eventually get something that works. Maybe not every time, not at all.
So are you saying maybe through this process that you're talking about, we'll get a pre-existing endogenous retrovirus that functions in a beneficial way?
Yes.
Well, it's a just so story. What we see today is these pre-existing endogenous retroviruses in the koala genome. They have the functional ability to disrupt the process of retroviral invasion. That's what we see. Pre-existing fixed ERVs disrupting the endogenization process of infectious retroviruses in the koala genome. So again, we have the fixed pre-existing ones that are functional and beneficial, and the ones that are newly endogenized or just exogenous retroviruses that are infecting. It's the pre-existing ones that are working to fight them off. I don't see how that helps your case.
But you still have an insertion, because you still have that insertion.
I've admitted
if
there's
you disrupt those sequences, you will no longer have functioning viral proteins from that, but you still have the insertion, right?
But it's disabled, so the insertion is going to be there, but it's not going to be of harm to the organism. That's the point. The pre-existing ERV sequences are acting in fighting off the harmful effects of infectious retroviruses. So when they disable them, yes, now they're disabled. They're no longer harmful, and probably over time, selection will remove this junk. And that's another problem I have with your model. Is in my opening statement, I covered the fact that there's significant evidence for genome-wide activity, let's say between 60 and 80%. But your side, and I'm not sure, I don't want to put words in your mouth, Luca, but from engaging your side, you guys want to say, well, most of that activity is just noise or it's spurious. But why in the world, over millions of years of selection, why would evolutionary processes keep all of that junk around if it's just noise? Because all it's going to do is muck up cellular systems. It's just going to be wasteful of energy and resources. What's your position on that, Luca?
To be fair, I just think that scientists, time to time, just have a very bad sense of humor. To call that DNA junk, and that was not the right call. I just think that scientists are blissfully unaware of this discussion, so they do very peculiar stuff like calling some chromosome Eve or things like that. They just don't care.
So how much would you
And
say then? So are you saying that this activity, this biochemical activity, all of this evidence for function in these non-coding regions of the genome in terms of regulation expression, is it your position that this is useful activity and not just spurious and noise?
I think that if you keep that unit of DNA, something must do. Of course, you will have dangerous part of DNA or things like that. But if you are functioning, you need to put that DNA to some use because you need energy to replicate that DNA and things
Right.
like that. So,
Well, and that's the problem with those that want to say that this is mostly just noise. And it sounds like from your position, you're saying that it's probably not just noise, because again, the energy requirements, a lot of energy is required. It's an intensive process to build that RNA chain. And so it wouldn't really make, but plus there should be evolutionary mechanisms that are suppressing all of this junk. But if it's your position that it is mostly active and there for a reason, then don't you
I
see how
want
that contradicts your
to
model? Yeah, go
say
ahead.
a couple of things. First
Yeah.
of all, yeah, but I think that organisms like mammals and us have a more refined genome because we do have a lot of mechanisms to prevent some errors and things like that. But if you look to a genome of an amoeba, you will find a very huge genome for a very small animal. Now, I want to ask you a thing because I think we are almost at the end of the
Yeah, I was just
discussion.
gonna hold. Don't lose your thought. I was just gonna say we have about just under 8 minutes left. So if you guys wanted to stay
Good.
on topic or try
I
something
want
new,
to
then
ask you a particular thing
sure, go
about
ahead.
widget.
About
So you were telling me that we do have evidence for widgets because I did not say anywhere that. To be clear, if we look at genome from humans near the flood, we would see those pre-viral elements. We do have example for that.
So you're saying at the time of the flood, with the archetypes, that would have been more heterozygous at that time than today, since there's been shifts in heterozygosity to homozygosity, a loss in some of these functional DNA elements. Are you asking for evidence for organisms in the past having more of these ERV sequences?
No, it's a different thing. If you read the paper, it was telling that in the past, in theory, of course, it's not a theory, it's an hypothesis, but let me slide that. We would see those elements, pre-viral elements that originated viruses afterwards. So we do have evidence for these elements not degraded and evidence for a perfect, let's say that, genome, because if your view is correct, we would be able to find it, because even pre-flood humans, we are talking about an event 4,000 and something years ago. So DNA, it would be not degraded at all.
Okay, so we accumulate in humans accumulate roughly 100 new mutations per person per generation. The mutation rate is quite fast in Y chromosome, mitochondrial DNA, biparentally inherited DNA. Within our DNA is ERV sequences, and they're also subject to mutations. They're subject to a lot of nearly neutral mutations. So even your ERV sequences that function in the embryo, for example, although that's essential, if they're hit with an effectively neutral mutation, it is harmful in the same way that, rust on the engine of a car isn't just going to destroy the car right away. It builds up. So yeah, we do have mutations that are building up in the genome and building up in pre-existing ERV sequences. So the genomes we sequence today, the ERV sequences that we see today, they have been hit with a lot of mutations. So they are a lot more degraded than, say, 4,500 years ago. But I don't know how you expect me to show you the genetics of humans right after the flood, where they have ERV sequences that have less mutations than today, where they're more in a pristine state. It sounds like you're asking me to show you an example of an original ERV sequence that was front-loaded and therefore had no mutations. Is that what you're asking to see?
Yes, and for what I can tell, it's quite easy. Find a corpse of someone really close to Noah or his sons, analyze his DNA, and find evidence for
Yeah, but
these elements.
yeah, but look at the examples we have of ancient DNA exist in what? Denisovans, Neanderthals. Neanderthals, for example, were the most inbred people group that ever existed. They have these massive stretches of identical letters, which means they have these massive stretches of what's called homozygosity. They were 40% less fit than modern humans. They accumulated a lot of deleterious mutations because of their environment, because of inbreeding. And so if I'm going to show you examples from ancient DNA, well, Neanderthals aren't going to help, since they were highly inbred and mutated and degenerate. So I don't think we have any examples of ancient DNA from non-inbred groups like Neanderthals.
But there is no reason to such a findings to be impossible.
Oh yeah,
We
no,
know
possible.
for sure that the son of Noah lived to be 300. So it should be possible to find such a sample or not.
Well, see, here's the thing. The ancient DNA that we're most likely to retrieve is going to be from some of these side branches that lived in the appropriate conditions that can preserve the DNA enough where it can even be reliable in terms of sequencing. And that seems to be the case with Neanderthals, because all the genetic sequences that we have seem to nest within that Neanderthal group. But again, that Neanderthal group was highly degenerated. So if we had an example of an early Neanderthal, or we had an example of, let's say, Heidelbergensis maybe, that may have been less degenerated, then we may be able to sequence that genome if we had it and determine, here's some ERV sequences that probably existed in them that don't exist today, because we've lost a lot of diversity, right? So that is a good question. But before time runs out, if I could, Luke, I did want to ask you a question here that applies to the Vige hypothesis, variation-inducing genetic element hypothesis. So I have a number of articles and papers where the secular community, for example, here, where did viruses come from? Let's talk science. Viruses might have come from broken pieces of genetic material inside early cells. These pieces were able to escape their original organism and infect another cell. In this way, we evolved into viruses. So essentially, we have the escape hypothesis from the evolutionary community that's saying, yes, a lot of these viruses may have actually come from cells, may have actually come from the genome rather than from outside. So are you familiar with this model? Are you, what are your thoughts on that? Go ahead.
Yes, it's an hypothesis, and it can be also true, of course, but we need to remember that we can also get things like protocells from experiments like the one done by David Dreamer. So it's not the only hypothesis. It's possible to get that from pieces of cells. Yes, potentially, if you have a cell broken down. A virus is a very, really simple structure. It has an envelope and a
Well, Luca,
string of
if I could,
RNA or DNA inside it.
if I could, because you're criticizing the Vige hypothesis. I've seen other evolutionary biologists as well criticize the Vige hypothesis, but we have in the secular literature, right? Here's a paper: Viral Evolution, Primordial Cellular Origins and Late Adaptation to Parasitism. So we have these evolutionists on YouTube that are criticizing the Vige hypothesis, saying that that's crazy. But we actually have this seen in your own secular literature, and they're even pointing out, for example, here's a question I have for you. The virus first hypothesis states that viruses predated cells and contributed to the rise of cellular life. Okay, this is not a young Earth creation source, of course. Contrastingly, all known viruses need a cellular host to replicate, thus necessitating the existence of cells before virus survival. That's exactly what we're saying in terms of our model, right? Obviously, there's some differences because I would look to the pre-existing fixed functional ERVs as being created units of DNA function, and then the unfixed ERVs that are harmful, disease-causing, and non-functional. I'd see those as exogenous retroviruses that actually did integrate, okay, through vertical transmission. But my question to you is, how do you address the problems that are put forth in these secular papers that say, yes, the best case scenario or a good case scenario is that retroviruses came from within the genome, since retroviruses require a host to replicate in the first place? So how do you address that issue?
First of all, there is a difference.
Hold on, Luca. Before you answer that, I just want to let you guys know that we are out of time. It doesn't matter to me. I don't know how punctual you want to be. So, of course, Luca, you'll be able to answer that. And then if you guys want to maybe just wrap it up in a few more minutes.
So let's be quick. Those are two hypotheses. First of all, it's quite different from the Vige hypothesis. And I said that the Vige hypothesis could be good if you bring evidence for it. I was not criticizing that much. I have other problems with the paper, but I do not think we have.
Well, Luca, and I just want you to answer this question as time runs out. One of the best lines of evidence is the same observation that the secular papers are pointing out, that retroviruses require a host to replicate. And therefore the question is, what came first, the host or the retrovirus? Well, obviously the host. This fits our model. God front loads the original created kinds with pre-existing functional DNA differences, genetic diversity, and variation-inducing genetic elements. And from ERVs, lines, signs, okay, we get RNA viruses that essentially are bad, harmful. And then eventually, they can move around the genome, causing harm, or then they can cross species, and bam, you got a fully infectious retrovirus. So one of the best lines of evidence is that fact. So if you want to reject the Vige hypothesis, how do you address that basic question? The basic question is, how can retroviruses originate from outside if retroviruses require a host to replicate in the first place? So just that question, I want to hear you answer.
Yes, as I said, we did observe in experiment from David Dreamer the formation of protocells with gene expression. So it's not the only possibility. We can have a form of life without a cell, and we can have an adaptation after that. Or
okay, so are you assuming that this protocell, through further adaptive episodes over time, you'll get a retrovirus?
Yeah, it will be possible. Why not?
Luca, and I mean this in all fun. I just want to put this full screen. This, I find every answer that you've given tonight is just a lot of story time, and because here's the thing, truth is important. And the evolutionary community and the YouTube atheists, they just want to keep saying ERV sequences, the best evidence for evolution. But yet, after debate after debate after debate, and you've even got, PhD scientists admitting, no, we have to assume over millions of years through maybe protocells, we get retroviruses, or somehow co-option takes place, and now an ERV sequence. Without it, we couldn't exist. I mean, I don't personally see why you find that to be convincing. It's just a lot of story time to me. And I don't mean that to be insulting. I just believe truth is important. And these ERV sequences function in so many incredible ways. We didn't even touch on a fraction of their amazing functional roles. For example, their role in tumor suppression, where through viral mimicry, which requires their sequence similarity to exogenous retroviruses, they can be turned on and the immune system can target that cell with more destruction and kill a tumor, essentially, through the help of what's called the P53 protein, which is the guardian of the cell. It's this relationship where they work together. I mean, that this is some amazing, you keep asking for evidence, and it's like, I don't know what more evidence are you looking for? Luca, take your time. Thoughts on that?
Okay. As I said, we know that we can get to very simple. They are not organisms because protocells are not that. But with abiogenesis, you will not start from a cell. You will start from something simpler. So either is possible. We can have very simple organism evolving at the same time, some adapting to be viruses, other to be cells, or we can actually have what you said, but they are both hypotheses, are not facts. But you cannot rule out one of the two.
And Luca, I want to be respectful. We'll give you the last word on the discussion. And if it's okay with our awesome moderator and host tonight, Sam, we can get into some closing statements. Luca, I do want to thank you for engaging in this important discussion. I also appreciate the prep that you've done, also for reading my book, bringing some good questions for us to discuss. Of course, it gets passionate at times, but you and I are good friends. And
Yes.
Again, I thank you for your presence in the debate community. So never any hard feelings, my good man. Very, very enjoyable discussion.
None taken. Don't worry about it. It was a pleasure to be here. So I'm more than happy for this debate. Let's get to the questions.
All right.
I think 5-minute closing statements. Luca, you can go first.
Sorry. It's very late from here. So I think that Donny did a very good debate. It did point out a lot of interesting things, but I'm still convinced about my positions. I think that I have very important questions without answer. The beach one was a very important one because if it's possible to find those units of not degraded DNA will be great and will be a very important evidence for this kind of things. And for your model, it's possible, and I want to see more research on that. And I cannot understand why we do not see that. Or maybe I can. So, very good debate. I will have to study a lot on these things. I did get pushed around, so I did not have any citation, any quote for what I said tonight, and it's a problem for a scientist. I hope to get that soon, and I can stop there.
All right. So thanks a lot, Luca. I just want to remind everyone to hit that thumbs up. It really helps with the algorithm. And Donny, brother, are you ready?
Yes. Let me start the timer.
All right.
All right, here we go. I could talk about this topic all day. I just put out a 2+ hour video presentation from a few days ago, just talking nonstop on ERVs. I mean, ERVs turn out to be some of the best evidence for separate ancestry and independent origins. And I find it hilarious because the evolutionists that want to, prop up shared ERV sequences as the best evidence for evolution turns out to be some of the best evidence for separate ancestry. And the ones making the videos, for some reason, shy away from debate. Stated clearly, Pelagia, Vice Rhino. I mean, bring it on. Because these guys understand that they can't address the amazing functionality of these ERV sequences. I mean, some of these ERV sequences, like HERV-H, also appear to have a responsibility in influencing heart cell development. And HERV-K helps embryos develop a built-in immune system that keeps them secure, okay, while the growing baby doesn't really have any antibodies to protect against pathogens from the outside world. So again, like, these are not just basic functions that you can just snip out and the organism still exists. No, like, literally without them, we couldn't exist. And the evolutionists, and I appreciate Luca engaging in this debate, because it's nothing against him. It's not his fault that he can't show us an example of a non-functional endogenous retroviral like element going from non-functional to functional, or altering its function to the point where it's now essential and critical for life to exist. Because PhD evolutionary biologists, the most militant ones who I don't even have to name, admit, no, we don't have that example. That they have to infer, and then they have to downplay it. They have to say, well, no, it's not about the function. And the reason why they got to say that is because they can't address the function. They have a genetics problem. They like to say we have a heat problem. They have a genetics problem. Okay. And so they want to point to these so-called mutational discrepancies within the ERV sequence and say, that's what matters. And then still not understanding that if the genome really is mostly functional, that means there's more hot spots than they've assumed, because they'll even admit, I've got paper after paper here where we have non-random mutations and hotspots. Okay, we also find that these functions were unexpected, because they'll like to say, well, these are expected functions. Oh, really? Because paper after paper says that they're unexpected. Anyways, unexpected findings on mutations. Okay, where we have these mutational hotspots, and in separate lineages, we have mutations accumulating that appear to reflect ancestry when in fact they were experienced independently. Okay, so the evidence for biological noise doesn't exist, and so it doesn't work. So I'm glad that Luca seemed to acknowledge that, which a lot of evolutionists are going to disagree with that, because they understand they need junk. They need junk, but junk DNA has been overturned. Transcribing a portion of the genome is a very energy intensive process. Every time a nucleotide is being added to a growing RNA chain, you are consuming a great deal of energy. These transcripts would just clutter up the interior of the cell if they were not playing any real operational role. We understand that a lot of these ERV sequences are only functional in certain developmental windows, and then they're turned off, and they can maybe be called upon again during the life of the organism for adaptive purposes, because DNA methylation keeps a lot of them silenced. Kind of like in your car, you have a backup camera, you have airbags, you have lane control, you have these built-in mechanisms that are not all turned on. So to the evolutionists, they see the backup camera that's not turned on or being used as apparently junk. It's just not being turned on until you need it. A lot of these ERV sequences are not expressed, or they're silenced until they need to be turned on. It doesn't mean they're junk. Okay, just like certain non-coding RNA genes, like pseudogenes, are only expressed under certain conditions. So a lot of these arguments are unsophisticated, and I do thank Luca for at least acknowledging this part. But unfortunately, acknowledging the fact that most of the genome activity really is useful, okay, also forces one to recognize the fact that there's more hot spots in the genome. So just like certain parts of a vehicle are prone to breaking down, okay, windshield and wiper blades, brake pads and rotors, battery, suspension, so on and so forth, more parts of our genome are susceptible to breaking down. So most of the genome's functional. There's more hot spots. The mutation rate is really, really fast, more fast than the evolutionists ever assumed. So yeah, we would expect similar mutational discrepancies between, let's say, humans and chimpanzees in the ERV sequence. But again, we didn't even begin to flesh out my hypothesis that results in predictions in the book that I wrote here that talks about how a lot of these so-called mutations are not even mutations. Because it's the evolutionist that assumes that all DNA diversity and all DNA differences are the result of mutations. But we would suggest that the majority of DNA differences are the result of design diversity. And a direct prediction of that model is DNA function. And we now have a trajectory that suggests genome-wide functionality. And this includes these ERV sequences, because with the overturning of junk DNA comes the overturning of ERV sequences as evidence for common descent. And I yield.
All right, what a debate. Good job, guys. So, Donnie, brother, I got, when I booted myself out of the party, I lost the first few comments that I placed in the private chat. So if you don't mind grabbing those, I can't see those anymore. And yeah, I want to also thank everyone for the super chats. You guys are awesome. I had a few super chats come in, and some gifts also. Doki, you're the man. Thank you very much.
Okay, so am I just going to go to the, okay, so here's the first couple questions that came in. I'll put them here. Oh, there you go. You can start with that one.
Yeah, all right. So Jay asks for $20. He says, please ask Luca which vestigial organs evolved first. We have 10: the heart, lungs, skin, intestinal linings, pancreas, kidneys, brain, liver, stomach.
Well, the first one was the digestive tract. On Earth, you don't need it if you are small. You don't need a blood vessel to just like look at something like insects. The concept is they are vital organs now, but if you look into more primitive organism, more simple organism, you will find that there is a progression with that. So you will have first a digestive tract. You will breathe through basically atmosphere because you are so little, and then you will start to have blood, but you don't need blood vessel because you are small, so you just need liquid going around, and then you need blood vessel because you are bigger, and then something to pump that blood through the vessel, so an earth, and so on and so forth.
So I guess my explanation is yes, I do find this to be a problem. Since the topic is ERVs, I'll give a response in the context of ERVs. And how this applies is the fact that many of these ERV sequences, right now, a lot of them are redundant. And a lot of them, we just have solo LTRs, which just represent functional stretches of DNA. Okay. And so, yeah, we do have a set redundancy in terms of the ERV. So even if they were snipped out, okay, you know, I can be born with both my hands missing. That doesn't really affect my fitness because I could still have kids, right? It's better if I have both my hands. But again, a lot of these ERV sequences could essentially be knocked out and the organism would still survive. Not ideal, but they could still survive. But many of them, especially the ones that are involved in embryological development, where if we didn't have them, we couldn't exist, these herb elements, or any of the functional ones for that matter, could not come about through slow evolutionary processes. That's why I say it's a lot of storytelling. That's why I like to respond to a lot of the evolutionists in terms of how they address the question about herb function with, cool story, bro, because that's all it is. And therefore it's pseudoscience. But again, many of these herbs are critical to life, but they're critical through irreducibly complex associations, okay, with other processes that's going on in the cell, because they're tightly controlled and they're integrated, that without their specific design and the processes that allow them to function in, let's say, the embryo, again, this is a critical design that we couldn't exist without. So my point is, yeah, there is a irreducible complexity even in the herb elements, because they couldn't be built up through slow evolutionary processes. Just like here, when it comes to the organs, the hearts, the lungs, the skin, the intestine, it all needs to be there. It all needs to be working from the start. Just like the Bible says, everything was created very good. And that's what we see with the human body. So go ahead.
Luca, question was for you.
Yes. I'm just adding that you can see those kind of things right now because we have organisms today with those characteristics. So maybe some does not have earth, some does not have lungs or things like that, and you can get progression from existing organisms. So it's not a story. We can see that right now.
All right, so we have another question from SWE for $10. She says, Donnie, isn't it the case that about 65% of the human and chimp Ys are shared? This leaves about 10 MB of Y sequence that is unique to each species. 10 MB is about 0.33%.
Okay, so good question. And I wonder, in my second PowerPoint presentation with another 500 slides, I've got some stuff on the Y chromosome, but I don't know if I'm going to be able to find it in time. So, yeah, good question. Okay, so I'll answer it here, and I think I can find it in time. I will share screen, Y chromosome, dissimilarity, essentially. Okay, let's see here. So when it comes to, let's see if I can get it, Y chromosome, within humans, there's an incredibly low variation. Okay, every single male Y chromosome, nearly identical, right here. The human Y chromosome exhibits surprisingly low levels of genetic diversity. Okay.
Humans. Can you go full screen if you're going to share that? Because the text is small.
Yeah, I'm looking for one. Okay, here it is. I'll go full screen here. Okay. So when it comes to, and good question, when it comes to the separate ancestry model, we like to look to the uniparentally inherited DNA compartments because essentially they're non-recombining. They're less messy, okay, than your biparentally inherited DNA compartment, your autosomal DNA. So we can build some really nice family trees. And again, there's low genetic diversity, but yet humans, when compared to chimpanzees, notice this paper, chimpanzee and human Y chromosomes are remarkably divergent in structure and gene content to the point where the human Y and the gorilla Y are more similar in overall architecture, gene content, and size differences. If you actually consider size differences, since the Y is half the size between humans and chimpanzees, it's only about 35% the same. If you don't account for size differences, it's about 70%. Either way, it's too far different because, again, the evolutionists will want to say, well, the Y chromosome is the most mutating, it's the fastest mutating chromosome. But wait a minute. If the human Y chromosome is mutating fast, which it is, it's about 3 per generation, why do humans overall have such low variation, 99.99% the same? We should find some highly divergent subpopulations if that's true, because we do have geographically specific Y chromosome, because after the Tower of Babel, certain groups spread out, they start accumulating mutations independently, but yet we're all still nearly identical. But then the chimpanzee, which is supposedly our closest cousin, is so vastly different and then also so different from the gorilla, which again is just one line of evidence that suggests separate ancestry. Yeah, the reason why the Y chromosomes are so different is because humans are separate in terms of the phylogenetic tree of life than chimpanzees. We're not related. And then they want to say, well, you know, it's the polygamous nature of chimpanzees. They're the odd man out, they'll say. You know, sperm competition has led to the chimpanzee's Y chromosome drastically changing, and the gorilla and the human Y chromosome not changing as much. So it's the chimpanzee that's the odd man out. But still, again, story time. Okay, here is the implications. What are the implications of this new information? First, for the evolutionists, the Y chromosomes must be evolving much more quickly than anyone imagined previously. Okay, here's the last thing I want to say is you can look to sperm competition, polygamous relationships in the chimpanzee, fast mutation rates, faster rates of gene conversion, whatever. You know, all these explanations they give in these papers, and which they admit are just hypotheses. They still need to be tested for there to be an empirical answer. This is what they're going to have to do. They are now going to have to apply mathematical models to try to demonstrate how a sequence can change extremely rapidly, including, notice this, wholesale rearrangements of significant parts and the evolution of entire gene families in a relatively short amount of time, yet stay homogeneous within a species. Because again, the human Y chromosome nearly identical worldwide. So, you know, that's the last thing I want to say is I get it. They have their stories. They have their hypotheses. They need to be tested. We need to see mathematical models. So I yield there.
Okay.
Go ahead, Luca. You want to respond to that?
I think I'll pass on that because I'm not knowledgeable enough to give a good response to that. I do know that there is an hypothesis from the secular side, but I do not know nearly enough to speak about that.
All right, Donny?
No, that's good.
All
that's
right,
good.
cool. You have to throw up.
So I've got a question. I'm not sure if you got this. Was this question lost too? Let me get it up here.
Well, I'm not able to put them in the chat from StreamYard anyway.
Okay.
So yeah, I believe that's right about where. So Mark asked a question for Donny. So how do you explain that the genome of modern humans is 1.5%, which is around 99 million base pairs longer than the Neanderthals and Denisovans?
Right. So I did tell Mark that I would debate him on this. So I'm getting all the very thorough technical questions, eh? Okay, so Neanderthals, essentially, why are Neanderthal and Denisovan genomes different than the human genomes, human genetics? Because phylogenetically, the way that the genetics are represented on a tree, the evolutionists want to say, well, you know, Neanderthals are a sister species, okay? And firstly, I want to say, when comparing the percentage similarity, we're all 99.999% similar. If you bring in the Neanderthal, we're about 99.7% similar to the Neanderthal. So we're still pretty similar. But what explains the differences, including the size differences? Well, a few things. Okay, a few things that come to mind, and then I want to touch on hypermutation being a reality. Neanderthals were the most inbred. We talked about this in the debate. They're the most inbred people group that ever exists on the planet. I got paper after paper in this slideshow. For sake of time, I won't pull them up. They were 40% less fit. They accumulated a lot of deleterious alleles because they had a lot of recessive mutations that came to the forefront. Since they existed in isolated small populations, they were inbred, which leads to rapid fixation of deleterious alleles. Okay, this is the end stage Neanderthal we're looking at. But Neanderthals in general were closer to the flood. So even before they became highly inbred and therefore had high levels of homozygosity versus heterozygosity, being close to the flood means they would have started off different. They would have started off with a different set of biodiversity. Okay. There's also a concept called patriarchal drive, where in the Y chromosome, you would be accumulating more mutations. Okay. Males would be accumulating more mutations, essentially, because if they're living hundreds of years old, that's more time for mutations to accumulate. in both the somatic cell lines, germ cell lines, and therefore more that are passed on. And so you would have differences there, because if Neanderthal was founded by an early biblical patriarch in his old age, he would start off with a whole bunch of mutations. And so on a phylogenetic tree, a long branch, which is assumed by the evolutionists to be reflective of deep time, could just reflect more mutations in the same amount of time. Okay, also because of the conditions they lived in, and we understand today, there's papers today where, notice this, 2 families had genetic drivers of germline hypermutation. So real-time examples today of people groups that were hypermutating, with some of them being hypermutated because of environment, but other groups being hypermutated because of, notice this, our findings imply that defects in DNA repair genes can increase what? Germline mutation rates. So that means a DNA repair system, we have multiple, over 10, DNA repair systems that are damaged and defected due to mutations result in more mutations accumulating from generation to generation. And in light of what we know about Neanderthals being the most inbred people group that ever lived, the environment that they lived in, I think it is very appropriate to extrapolate that to mean that Neanderthals had mutations in many of their important DNA repair enzymes, which means what? More mutations are accumulating, more mutations are becoming fixed, explaining exactly why Neanderthals are different to modern humans today. So that's about 3 hours worth of information in a few minute answer. So go ahead, Luca, if you had anything you wanted to add.
Yes, of course, I do not know what's making the difference, but we do have DNA from those humanoids, from Neanderthals, Denisovans, and us. So I think we can get what happened here. And if you did the question, I think you know better than me. It's peculiar that things like Neanderthal and the Denisovans are kept out of the work of someone like Jensen, and I find that fascinating. I will see. I would. I would love to see how the last common ancestor date will turn out if you do that. But apart from that, maybe it was some mutilations. Maybe we are looking at duplication or insertion of other elements like transposon. I do not know.
I guess I'll do a real quick final word, Sam. I think you're on mute, brother. I just
Yeah,
wanted to
go
pull
ahead.
up some of these papers. Inbred Neanderthals left humans a genetic burden. So 40% less reproductively fit than modern humans. So they lost a lot of their genetic diversity. They would have started off with higher levels of genetic diversity, being closer to the flood, the early Neanderthal, okay? Because the DNA we have today is of the end stage Neanderthal, when they were at their worst genetically, so genotypically and phenotypically. We understand that they were cold adapted, right? So the way that they had prominent brow ridges above their eyes, dominated by a very big wide nose, strong muscular bodies, wide hips and shoulders. They were adapted for the cold ecoglacial environments that they existed in. But over time, due to their isolation, their inbreeding, they would have lost a lot of genetic diversity. Okay, they would have accumulated a lot of harmful mutations. And there's paper after paper in the secular literature on this. Notice this, the genetic cost of Neanderthal introgression. Recent studies have shown that this Neanderthal DNA is depleted around functional genomic regions. This has been suggested to be a consequence of harmful epistatic interactions between human and Neanderthal alleles. So if anyone's interested, I did a 3-hour presentation on Neanderthals a few months back where I back up everything I'm saying with the secular literature. So I yield.
All right. So next we have a question from Doki. He's asking you, Donnie. He said, "Is your closet full of nothing but blue shirts?"
If you open my closet, It's just a big lineup of SFT blue shirts. One for Monday, Tuesday, Wednesday, Thursday, all the way. But Sunday, I'll dress up. Because by Sunday, my wife's sick of looking at me in this shirt, right? At least I can do.
All right. So next we have a problem, unless you wanted to respond to that, Luca.
And I have just shirts, so
All right. Looks good.
no blue here.
All right. So we have a question from Obamacare. He says, question for Luca. So they're coming at you now. He says, can you provide an example of a beneficial mutation, I guess, in the context of this discussion?
Well, we are talking about transposons, so maybe not mutation, but the one in maize, corn, they were quite good. Maize is a stronger, better plant. That's one. Also, let me think. The mutation we find in my area that reduce the probability to die from earth attack. Sorry, yes, that's two.
I can respond. So beneficial mutations. I've got some slides here from my study into beneficial mutations. And here's, again, the mutation rate you can see here. Mutation and human exceptionalism or future genetic load, Michael Lynch. An average newborn contains roughly 100 new mutations per person per generation. And they admit that most of these mutations that accumulate are effectively neutral, meaning they're very slightly deleterious. Okay, so they accumulate, they're effectively neutral, which means they're slightly deleterious, like single rust molecules on a car or a single spelling mistake in a book the size of an encyclopedia. That means they're invisible to selection. Okay, selection can't see neutral mutations, but what we know now, especially about the function of the genome, is it's not appropriate or accurate to say neutral. It's more appropriate to say effectively neutral or low impact or nearly neutral. Because notice this. It seems unlikely that any mutation is truly neutral in the sense that it has no effect on fitness. All mutations must have some effect, even if that effect is vanishingly small. Okay, so here's the thing about beneficial mutations then. Most beneficial mutations that we study on a molecular level, okay, because oftentimes what we'll do is we'll look at the phenotype, the new variation. But what we should be looking at is the genotype and seeing what's going on a genotypic level. And what we'll find is a lot of these examples, they're reductive. It leaves the organism functionally compromised. It's a reduction that's leading to a temporary advantage, like sickle cell anemia. It leads to a type of resistance, but it's actually due to a reduction because it's due to a broken cell, broken protein, okay, a deformed hemoglobin protein, essentially. And that means there's no way to counterbalance the damage due to the influx of so many low impact deleterious mutations that are accumulating. We also have a lot of examples of the variation and phenotypic novelty based on epigenetic means. So we have a lot of latent information like these retrotransposons that are now manifested. They're pre-existing, they're manifested, they lead to change, novel change, but it's not the type of change that large-scale evolution needs because it's already built into the system. There's a lot of morphological adaptability built into the system. Even his example of the lizards. We understand that some of these lizard species have dual reproductive mode capabilities, where depending on the environment, one mode is emphasized over the other. And if one mode is emphasized over the other for long enough periods of time, the other one will reduce and eventually it'll be lost. So we're not seeing the adding of any true novelty. We just see the expression of pre-existing variation that's already built into the system. And here's what I typically say, because I mean, we could talk about all day, so-called beneficial mutations, but they can have 100 of them. They would need millions of truly beneficial mutations to counterbalance the damage done by the pouring in of so many damaging mutations. So That's my brief thoughts on beneficial mutations. Luca, you can have the last word. It was your question. Oh, I think Luca's
You're on
muted.
mute, Luca.
The example I gave in the debate with Ken were quite good. We have cows, and a lot of people asked me why it's beneficial to the cow. Yeah, it's beneficial because that cow will be selected to reproduce. So it's more fit to survive in that way. And it's adding things because you are producing more, you have more tissue, you are more blood vessel. Maize, it's a better plant, bigger plant, stronger plant. I do not see anything worse there. And also those were not deletion. We have also duplication and things like that. In us, for the most part, are harmful. But when you got a good one, it will be selected. The quantity of mutation does not mean anything if any bad mutation will eventually be selected out and the good ones will be kept.
All right. So the next question is from you, or for you, from Doki, Luca. He says, or he asks, did the RNA world give rise to the DNA and eventually ERVs?
Allora, sorry, I'm that tired that I'm starting to talk in Italian. So, yeah, the difference between the two is a sugar. I do not know the exact process, but we know for sure that RNA and DNA share almost the same basis. So I can see a process where you can form DNA from RNA. I do not know the details, but you just need to have a a nucleotide, and then to form the chain, and that could be helped by the RNA. I think that RNA molecule was the first to be formed, and then DNA.
Yeah, so you think the ERVs derived from them?
No. At that stage, you just have RNA. I do not think you have ERVs at that stage.
What do you think about that?
Okay, so the question, did the RNA world give rise to DNA and eventually ERVs? So again, I think especially when it comes to the chicken and egg problems with abiogenesis, and the fact that design diversity seems to better explain variation in the biological world. When it comes to retroviruses, I believe the scientific data is best supported by the fact that retroviruses, RNA viruses, would have originated from within the genome. And I think the Vige hypothesis, although there's a lot of work to do, which is what I'm doing, and I'm going to be elaborating on it in my follow-up book, the findings of the new biology demonstrate that mainstream scientists are wrong regarding the idea, because they want to say there's some kind of RNA world where maybe these precursors to retroviruses came about, and then eventually they adapted in a way where they are now required of a host to replicate. and that's, they understand that problem, right? They have multiple hypotheses, but they understand that's a big issue since retroviruses require a host to replicate. So it makes more sense, since there's kind of a conundrum there, a chicken and egg problem. What came first, the host or the virus? That the viruses came from the host. Okay. So those that want to reject that model have some problems, some hurdles, and stories aren't going to cut it. So Again, instead, RNA viruses originate from transposable elements that were designed as variation-inducing genetic elements, created units of DNA function, I like to call them, because they do so much more than just create variation. They're functional in all the different ways we talked about in the debate today. Due to the redundant character of these elements, their controlled regulation may have been readily deteriorated, and some of them may now merely cause havoc. Again, Darwinists are wrong in promoting ERVs as remnants of invasions of RNA viruses. It's the other way around. To date, no clear explanation. This is from the secular source, or origin of viruses. There's a lot of problems, and they readily admit it. No clear explanation for the origin of viruses exists. Viruses may have arisen. Honestly, that's exactly what I've been putting forth. Viruses may have arisen from mobile genetic elements that gain the ability to move between cells. Exactly. Okay. The host was created by God, and then the host was front loaded with pre-existing diversity and many classes of functional DNA elements like ERVs and transposable elements. And because of degeneration, because of recombination or errors in the packaging process, retroviruses came about. They can move between the cell, or they can cross species, and there you go. Fully infectious retrovirus. Last thing I'll say is it's fascinating because you can look at the genes of these retroviruses, their components, and actually track back, and that's some work that we're doing, is track back to what the original host was that it came from. HIV did not originate in humans. Started off as SIV in chimpanzees, and I believe which came from some kind of rodent species, which may have been its original host, where it was not harmful. These become harmful when they cross species, and now the species that it's crossed into cannot regulate the invading retrovirus. And so it burns hot and fast, leads to disease. I mean, this to me is the only way to explain it. So there we go. I yield.
Yes, to respond a little bit more in detail. So, for what I can recall, DNA needs an inorganic acid, the same of RNA, a slightly more complex sugar, but we are talking about one carbon more, nothing more than that. And bases are basically the same. So I do not see at this moment a reason where it would be impossible to have both. I think that the process to synthesize DNA was maybe more late than the RNA one, for what we can tell, but I see no reason to not have that compound.
All right. So Howard asks, he says, Luca, another question for you. He says, how can ERVs being functional BD predict by your model? That also said prior that ERVs being junk was predicted. So you understand the question? He's asking you how.
I would say give me a source for that, because if you have ARVs, you will have something that usually synthesize proteins for the virus. So it's quite simple to change something in that and get something else. I will not say that was the case. Maybe I'm wrong. I'm waiting for the sources.
So since this is the last question, I'll be really brief because we've hit the 2.5 hour mark and I'm pretty much out of energy for this. So yeah, the evolutionary community, they didn't even really predict. They expected some function, some regulation in the non-coding areas, but they did not expect or predict just how much function we would discover. This 60 to 80% evidence for activity, about 60% evidence for transcription, all this amazing evidence for function in pseudogenes, ERV sequences, ALU sequences, all these other classes of retrotransposons, to the point where they're essential, they're critical. Without them, we wouldn't exist. No, they didn't predict any of that. So you'll see some of these guys these militant critics, like there's one, Tony Reed out there. He says, these creationists, they don't know what they're talking about. We never said that there'd be no function. Yeah, you might have expected some in the non-coding regions, okay, but not nearly as much as we've discovered, which is why us as creationists, we've made some really fascinating future novel predictions, okay, that only future observations, testable predictions, the gold standard of science on DNA function. I've made a number of them in my book here, comparing fixed ERV sequences and unfixed. So I do recommend it. Book should be linked in the description box. And claiming that the majority of this activity is just noise or spurious, it doesn't work. It doesn't work for the numerous reasons that we've discussed tonight. So I yield.
All right. Well, I guess. Oh, you want to respond to that, Luca?
No. It's fine.
Yeah. All right. Well, then, hey, I want to thank everyone for showing up, and don't forget to hit that thumbs up. And again, there's Luca, your channel, I'm sure, is in the description.
Yes.
And yeah, I guess that's a wrap.
That's a wrap. Sam, we got to have you for more future debates, brother, because you did a fantastic job. Yeah. And Luca,
it's basically morning here, so I'm literally out.
I'm out too. Luca, I appreciate you doing this so late since we're on opposite times.
Yes, I made you sweet, and that's a good thing. That was a very good debate, I think.
It was very good. It was very good. I look forward to rewatching or watching technically for the first time. Sam, 2.5 hours. Our debates kind of go long, so thanks for giving us your time. John Maddox, the after show man himself, he says after show will start right after debate ends. So I did promise, even though, man, I need like an IV of coffee right here. I'm going to take probably 20 minutes off, get some food and a coffee, and then I'll join to battle it out with some of the evolutionists, I'm sure.
Need some sleep, but I think that I'll lurk a little over LPP. Sorry, Maddox, I will not join tonight. Sorry, I'm too destroyed to do that.
Next time.
Completely understand. Luca, we just debated the most technical topic in the creation evolution debate for almost 3 hours. So anyways, Luca, thank you. Sam, you're the man with the plan. Thank you so much, brother. And to the audience, thanks for joining. Standing for Truth is out.